Ever wondered how scientists can tell exactly how much caffeine is in your energy drink? Or how a pharmaceutical company ensures the purity of a life-saving drug? They're not tasting it, they're using light. And one of the most powerful ways they do this is with a technique that sounds complicated, but is beautifully simple. Welcome to our guide on UV-Vis spectroscopy, or as it's more commonly known, UV-Vis spectroscopy. In this video, we're going to demystify this essential engineering tool. You will learn, first, the fundamental principle of how molecules interact with light. Second, the famous Beer-Lambert Law, the simple equation that makes it all work. Third, what's happening inside the molecule, the fascinating world of electronic transitions. And finally, we'll look at the instrument itself, a spectrophotometer, and see why its modern design is so clever. Let's get started. First things first, what is UV-Vis spectroscopy? Formally, it is an analytical technique that measures the amount of ultraviolet or visible light absorbed by a substance in a solution. So, what does that really mean? Imagine you're shining a flashlight through a glass of juice. The light that comes out the other side is a bit dimmer, right? That's because the juice absorbs some of the light. UV-Vis spectroscopy is just a very precise version of this. We shine a beam of light of a specific color or wavelength through a sample, and we measure exactly how much of that light got blocked or absorbed. The more concentrated the juice, the more light it blocks. It's that intuitive. Here's how to picture it. We start with a light beam of a known intensity. It passes through our sample, which is held in a small transparent cell called a cuvette. A detector on the other side measures the intensity of the light that makes it through. The difference tells us how much light was absorbed by the sample. This simple principle is used everywhere. It's a workhorse in quality control labs to check the concentration of active ingredients in medicines. Environmental agencies use it to measure pollutants like nitrates or phosphates in water samples. It's a fast, cheap, and reliable way to find out how much of something you have. Now, you might be wondering, how do we connect the amount of light absorbed to the actual concentration? That brings us to the most important equation in this field. This relationship is described by the Beer-Lambert Law. It states that the absorbance of a solution is directly proportional to the concentration of the absorbing species and to the path length of the light through the solution. Let's go back to our juice analogy. The Beer-Lambert Law is just two common-sense ideas combined. First, there's Lambert's Law. It says if you have a wider glass of juice, it will block more light. That makes sense, right? The light has to travel through more stuff. This is the L in the equation for path length. Second, there's Beer's law. It says that if you have darker, more concentrated juice, it will also block more light. This is the C in the equation for concentration. The equation, A equals epsilon CL, just combines these ideas. A is for absorbance, a measure of how much light was blocked. And that little Greek letter, epsilon, is called molar absorptivity. Think of it as a constant that's unique to the substance itself. It's a measure of how good that specific molecule is at absorbing light at a specific wavelength. Visually, it's a straight line. If you plot absorbance on the y-axis and concentration on the x-axis for a series of samples, you should get a perfect straight line passing through the origin. This is called a calibration curve, and it's how we find the concentration of an unknown sample. One important warning, however, the Beer-Lambert Law works beautifully for dilute solutions. If the solution is too concentrated, the relationship breaks down and the line starts to curve. This is the core of quantitative analysis. Want to know the concentration of DNA in a biological sample? Measure its absorbance at 260 nanometers and use the Beer-Lambert Law. Need to verify the concentration of a drug in a batch? Same process. It turns a simple light measurement into a precise concentration value. So we know that molecules absorb light, but why? What is actually happening on a molecular level? The answer is electronic transitions. When a molecule absorbs UV or visible light, the energy from the light photon promotes an electron from a lower energy molecular orbital to a higher energy unoccupied molecular orbital. Think of the electrons in a molecule as living on different rungs of a ladder. The rungs represent the energy levels of the molecular orbitals. To jump from a low rung to a high rung, an electron needs the exact amount of energy. It can't just take half the energy and get stuck in between. A photon of light is a tiny pocket of energy. If a photon comes along with the perfect amount of energy to match the gap between two rungs, the electron will absorb it and make the jump. If the photon's energy doesn't match, it just passes right through. This is why a substance only absorbs specific colors or wavelengths of light. Engineers classify these jumps or transitions into four main types. The highest energy jump is from a sigma bonding orbital to a sigma anti-bonding orbital called a sigma to sigma star transition. The lowest energy jump is often from a non-bonding orbital to a pi anti-bonding orbital, an N to pi star transition. The type of transition a molecule can make determines whether it absorbs high-energy UV light or lower-energy visible light, which is what gives things their color. This concept explains why things have color. A carrot is orange because the beta-carotene molecule inside it strongly absorbs blue and green light, reflecting the orange and red light back to our eyes. Those absorptions are caused by, you guessed it, pi to pi star electronic transitions. This is also how we can identify substances. The specific wavelength a molecule absorbs, called its spectrum, acts like a unique molecular fingerprint. Let's quickly cover two terms you'll hear all the time. First, chromophore. The chromophore is the specific part of the molecule that is actually responsible for absorbing the light. Think of it as the molecule's color center. For example, a carbon-carbon double bond or a carbon-oxygen double bond are common chromophores. Next, we have an oxochrome. An oxochrome is a group of atoms attached to a chromophore which modifies its ability to absorb light. It doesn't cause the color itself, but it can shift or intensify it. Think of it like a tuning knob for the molecule's color. So, what machine does all of this work? A spectrophotometer. Let's look inside a modern one. The setup is brilliant. It starts with a light source that produces both UV and visible light. This light enters a device called a monochromator. Its job is to act like a prism, splitting the white light into all its individual colors or wavelengths and selecting only one very specific wavelength to pass through. Now for the clever part. A beam splitter, which is like a half-silvered mirror, splits the single wavelength beam into two identical beams. One beam goes through our sample cell. The other beam simultaneously goes through a reference cell, which contains only the pure solvent without our substance. Two separate detectors measure the light intensity coming from both the sample and the reference at the same time. Finally, the readout device compares the two signals and displays the final, corrected absorbance. Which brings us to a crucial question. Problem: why go through all the trouble of splitting the beam? Why is a double beam design better than a simpler single beam instrument? Let's walk through it. In a simpler single beam instrument, you have to do two separate measurements. First, you put in the reference cell, or blank, to tell the machine what 100% light transmission looks like. Then you physically take it out and put your sample in to get the final reading. But what if the light source flickers or its intensity drifts even slightly between those two measurements? The final reading will be inaccurate because the baseline you set is no longer valid. The double beam design solves this problem elegantly by splitting the beam and sending it through the reference and the sample at the exact same time. The instrument is constantly comparing them. If the lamp flickers, both beams flicker by the same amount at the same instant. The detectors see this, and the electronics calculate the ratio of the two signals. Because both signals change together, the ratio between them stays constant. This design automatically corrects for any fluctuations in the lamp intensity or detector response in real time. This provides a much more stable baseline and leads to far more accurate and reliable absorbance measurements. So let's quickly recap what we've learned. 1. UV spectroscopy is a technique that measures how much light a substance absorbs. 2. The Beer-Lambert Law, A equals epsilon CL, gives us a simple mathematical way to relate that absorbance directly to the substance's concentration. 3. This absorption happens because photons give electrons the exact amount of energy they need to jump to higher energy levels, a process called electronic transition. And 4. A modern double beam spectrophotometer provides highly accurate results by using a reference beam to correct for any instrument fluctuations in real time. And just like that, you understand the fundamentals of one of the most essential tools in modern science and engineering. If you found this explanation helpful, please hit the like button, subscribe for more videos that make complex engineering simple, and let us know in the comments if you have any questions. Thanks for watching.